Good morning. I would like to thank the organizer for inviting me to discuss my work on epigenetic regulation of myelination and remyelination. My name is Patrizia Casaccia, and I work both at the Advanced Science Research Center at CUNY and the Icahn School of Medicine at Mount Sinai. So for today, the goal would be to just start with a brief introduction, discussing the difference between genetic mutations and epigenetic modifications. And then we will go through 4 different objectives that I would like to review with you. And then we summarize and we'll draw some conclusions. Now, when we think of genetic mutation, we typically refer to alteration in the DNA sequence that we can detect in every cell and in every tissue of the body. Epigenetic mechanisms, in contrast, have a different flavor, and they are actually cell-specific mechanisms that do not necessarily require any change in the DNA sequence. So when we discuss in terms of epigenetic mechanism, the modality of regulation of gene expression include histone modification, DNA methylation, also association to the nuclear membrane or microRNAs, as depicted here. And we will review each of these modifications, starting with the first objective, review the work relating to post-translational modification of histone proteins occurring during oligodendrocyte differentiation. Now, if we think of the DNA as wrapped around these core nucleosomes, there are octamers of protein called histones, and for each histones we have very long tails, each including a number of amino acids that are amenable to modification from the environmental stimuli based on the activity of specific enzymes. And more specifically, what have acquired a lot of interest are modification of arginine and lysine residues. And today, just for necessity of time, I will just focus on a very, very brief review of the work that we have published through the years, defining the histone marks and the enzymes responsible for those epigenetic modification in terms of both developmental myelination and adult myelin regeneration. Now, here we can detect the schematics of an oligodendrocyte progenitors, when in orange the nucleus of the cell. What you can see that at the progenitor cells corresponds a very defined appearance of the nucleus, which is really electrolucent, which underscores the importance of the dispersed chromatin that we find at this stage. And then as the cell progressively differentiates into a pre-myelinating oligodendrocyte and ultimately a myelinating oligodendrocyte, you can see how the appearance of the nucleus changes dramatically with progressive, more and more condensed appearance of the nuclear chromatin. This state, the progenitor state, characterized by what we call euchromatin, relaxed euchromatin, is characterized by acetylation and active gene transcription. This mark then is removed by histone deacetylases. Other marks are present in terms of repressive methylation on arginine or lysine residues, and this eventually leads to a repressive environment which morphologically is associated with this compaction, transcriptionally inactive chromatin. And then there are changes in the DNA methylation that we will review further. So the work of several years has allowed us to come up with this model of oligodendrocyte differentiation based on a progressive chromatin repressive landscape, which serves the purpose to decrease the expression of transcription factor that would inhibit myelin genes, neuronal genes, cell cycle genes, and chromatin modulators. And through the years, what we have come up is this model where if you would see this brain as the brain of a child, you would look inside and you would see that progenitor cells are highly migratory, proliferative cells. But if you go into the nucleus of the cells, there's a lot of activity ongoing. And you can appreciate here the DNA wrapped around these histones. And you can see here these little histone tails with the different enzymes depositing marks, being acetylation or methylation of specific groups, leading to this dispersed chromatin that I showed you before, very active environment, which allows the cells to be migratory and to be proliferative. Then eventually, though, many of those changes will need to be put to rest. Cells have to exit from the cell cycle. And this is achieved, for instance, by allowing those cells, by allowing those genes regulating a cell cycle to be really condensed and compressed. And this is defining then the features of the mature oligodendrocytes, which is quite, which is also the identity that we see in the adult brains. Now, what's interesting is then when we look then at this kind of marks in the brain of patients with multiple sclerosis, we actually have a defective deposition of this histone repressive mark, which is a characteristic feature that we can also detect in mice at an older age. So if we want to take home a message from this first objective, we can conclude that these repressive epigenetic marks, referring mostly to post-translational modification of histone tails, are necessary for developmental myelination, and that the deposition of these marks is impaired in postmortem MS tissue. Now, a second modification that I mentioned when I was reviewing the epigenetic modifications is the role of DNA methylation. And DNA methylation, catalyzed by the DNA methyltransferases, in this case DNMT1 or DNMT3A, are defined by the deposition of a methyl group on a cytosine. And this is characteristically linked to gene repression when the modification is especially present at the level of the promoter. So in 2016, when we start looking at several of these modifications over time, and we noticed that there is an increase of cells with a high level of DNA methylation that occurs during developmental myelination. We performed genome-wide approaches to define all the region of the genomes where this DNA methylation was occurring. And what we noticed is that there were several genes that were responsible for the mature myelinating state, including MAG, MOG, and the RG1, that were hypomethylated. So in other words, during the differentiation, we would have repression occurring on all those genes that are incompatible with the mature state. And we would have hypomethylation, so less DNA methylation, and therefore a de-repressive state for those genes that are important for the mature state. Now, if we knocked out DNMT1, a DNA methyltransferase is responsible for the deposition of these marks, you can see here in green myelin, it's almost completely gone from the spinal cord of those mice. And this is different from knocking out a similar isoform, which is DNMT3A. And you could see here, you could appreciate here, the phenotype of the mouse, which lacks DNMT1. You can see how it is severely ataxic and is shaking, and you can envision how this is possible when you look at the appearance of the spinal cord. It's almost translucent. There is very little myelin left here. You can also appreciate it here at the level of the brainstem, how the myelin that's present in the control is absent in the DNMT1. So from those studies, eventually these mice will die very early on. So this clearly showed us that DNMT1 and DNA methylation is necessary for the differentiation of those cells. Now, what's interesting is that in this case, however, and this is work from Ranjan Dutsal's lab, he could show that when he compares tissues, MS tissues from myelinated or demyelinated hippocampus, he could identify from MS brain, he could identify that those very same molecules, the DNA methyltransferases, are actually increased in the MS tissue. So, as take-home message of the second part, the DNA methylation catalyzed by the NMT1, we could show it was necessary for oligodendrocyte differentiation during developmental myelination, but that the level of the DNMTs, in this case, are elevated in MS tissue, which led to the question, what does DNA methylation do in MS tissue? Where is it localized? And more specifically, we were interested in understanding whether those epigenetic changes, not anymore in the lesion itself, but in the unlesioned white matter, could explain in part the disease process. And so this is a study that we published in 2014 when we could take control versus MS brain. And we were able to identify from this brain, we performed DNA and RNA isolation, and then we analyzed both this distribution genome-wide of the methylation on each chromosomes. And again, the take-home message for this study was that what we notice is that what's called even normal appearing white matter is quite distinct in the control and in the MS brain. And in the MS brain, we see increased expression of genes involved in antigen processing, but decreased expression, both in terms of transcript and in terms of proteins, of genes whose expression is regulated by DNA methylation. So in the follow-up studies from these initial results, we wanted to understand what was the function of those genes that we identify as hypermethylated and therefore repressed in the normal appearing white matter of patients. And this is one of the genes that I was mentioning to you before. It's called NDRG1. And this gene we find is being upregulated during oligodendrocyte differentiation, and yet this is one of the main targets for hypermethylation in MS brain. So here you can see how in normal differentiation, this gene is hypomethylated as progenitor cells become oligodendrocyte. Precisely the opposite of what happens in MS patients. So what do we know about these genes? We generated a reporter mouse. We can see that these genes is really expressed very nicely where MBP is identified. So this gene is in green, it's a reporter mouse. So you can see how there is almost superimposable expression with MBP. It's completely excluded from neuronal genes. And these genes, it's also interestingly decreased as demyelination ensues. So we knock down eventually this gene, and we notice that in the knockout, there is increased damage and decreased repair in the Cuprizon model of demyelination. So the take-home message of this third part is that in the unlesioned white matter areas of the MS brain, we detect higher expression of this DNA methyltransferases, as also consistent with what Ranjan had published. Higher DNA methylation level than controls, and this result in lower expression of genes that by functional characterization we identify as being critical for myelin formation and maintenance. So the conclusion is that the normal appearing white matter in this MS brain, which is characterized by increased DNA methylation, also renders oligodendrocytes in these regions more prone to injury. And finally, in the last few minutes, I just wanted to address with you the importance of this DNA modification during oligodendrocyte differentiation and aging. I mentioned to you before how many of the histone modification that we identified as necessary for developmental myelination were then reversed during the aging process, a situation that was quite similar to what we detected in the postmortem of brain of MS patients. And so since there is an epigenetic clock, which is characterized by increased DNA methylation over time, we really wanted to explore more the relationship between other also DNA modification occurring over time. So I mentioned to you previously the importance of post-translational modification, the importance of DNA methylation. I'd like now to introduce an additional modification. So the DNA methylation, which I discussed, catalyzed by the DNMTs, where the DNMTs are responsible for this deposition of methyl groups, is involved with gene repression. However, within the past few years, we have been able to identify a different modification, which is DNA hydroxymethylation. And the hydroxymethylation has a different consequence of DNA methylation because it's typically associated with high level of gene transcription. This is catalyzed by a number of enzymes that are called the 1011 translocases, the TET enzymes, which remove this methyl group from cytosine. They oxidize into this hydroxymethyl cytosine, which eventually leads to the removal of any modification and the availability of a cytosine. So since myelin regeneration declines with age, as depicted here in young adults, we have successful remyelination after an induced lesion caused by lysolecithin injection. In the older mice, the repair process is impaired. And it is also depicted here by seeing less fluoromyelin and fewer remyelinated axons over time. There is also this characteristic appearance of this extracellular space very close to the axon and between the axon and myelin in these older mice. So we wanted to know, we had identified the role of the DNMTs in developmental myelination. We really wanted to approach the importance of hydroxymethylation during the adult phase and in the aging. And so what we notice is that in young mice, during the repair process from a lysolecithin lesion, we do detect an increase of hydroxy DNA methylation in oligodendrocyte cells, a feature that we do not see in older mice. And when we look then at the level of the enzymes that are responsible for this, while they are pretty much quite elevated in young adults compared to the neonates, they decline dramatically with aging. And this is a characteristic that we see only for one isoform, TET1, not for TET2. So we generated conditional knockouts for each of these isoforms, TET2 and TET1. And really interestingly, TET2 had completely no effect, while the TET1 ablation in conditional knockout, on inducible conditional knockout, remarkably decreased the ability of those cells to regenerate myelin after lysolecithin injection. In addition, you may recall that I mentioned how in young mice, there is a very nice tight relationship between myelin and the axon, so that the axomyelinic space is really, really tight. And this is in contrast with what we detected in the old mice. So we asked what happens in the mice that are lacking, in young mice that are lacking the TET1. And what we detected is an expansion of this axomyelinic space, quite similarly to what we detect in older mice. So to further understand the gene target that could regulate this axomyelinic space, we performed a number of studies, and we detected an important solute carrier family members that are detected just here at the axomyelinic interspace, and that they are completely devoid of in these knockout mice. Now, what's interesting is that the solute carriers that are responsible for this regulation of the space are decreased over time, and therefore we infer that this is really an important factor in determining this appearance and this inefficient conduction of this remyelination in the older mice. Remarkably, these hydroxymethylation and TET level are decreased in MS tissue, thereby leading to the fact that it could be really important to follow up on some of these TET1 gene targets for putative therapeutic potential. So in summary, I have shown to you that in the DNA methylation and repressive histone post-translational modification are necessary for developmental myelination, and their inaccurate deposition occurs in MS brain, and also that DNA hydroxymethylation catalyzed by the TET family is necessary for myelin repair in young adult, There is a progressive age-dependent decrease and also a decreased expression in the MS brain. So from this is just a graphical abstract summary of what I mentioned before. I would like to just highlight the potential, perhaps, for rejuvenating strategies of the myelin regenerative capacities by harnessing our knowledge on the levels of TETS and the regulation and potential gene targets. And finally, I would like to thank the people in the lab that have performed this work, most relevantly Sarah Moyan and Damien Marshall, previous past members of the lab that have contributed to this work, and our collaborators and funding sources. Thank you.